article, but welcome to the first seminar of the new series. We have a pretty cool program this time, so we'll send out a newsletter in a short while where we kind of just summarize all the upcoming seminars. And as part of the cool program, we have a young investigator, because we're not only bringing in all the old fogies that you've seen at many ventures and stuff like that, we also try to really tease out the up-and-coming investigators in the liver field. And we have such a species with us today, and very excited to welcome Nataliya Razumilava. I hope my German accent kind of helps with the pronunciation of the Belarusian accent. So she's from Belarus originally. That's where she got her MD, but she did her residency in Detroit, Michigan, you know, one of the cool cities now in the United States. And she rose through the ranks there, and eventually, I think, did 2 fellowships, right? One in GI hepatology, one in transplant, right? And then also became really invested in research. I'll talk about that in a second. And then her clinical experience continued, becoming, I think, an assistant professor for a short time in Michigan, right? At Mayo, right. And now she's in Michigan and has built a pretty prolific program there. I think you're also in charge of liver tumor kind of program, right? Is that true? Co-in charge. Okay. Basically doing all the work and somebody else gets all the credit. And so I became aware of Nataliya's research because she was one of the few people that really started working on the biliary system when it wasn't as cool as it is now, you know. So now everybody realizes how important the biliary system is, but in the beginning it was a pretty tight-knit kind of community, and not too many people were working on it. And in particular, I was very impressed that she started looking at the extrahepatic bile duct, which really a few people have done, and so she's done some pretty important work in that space, and also in liver tumors, and we'll hear about that today, I believe. But her career is also a really nice example of how to kind of make it towards independence. So she got one of those Pinnacle Awards from the ASLD, you know, one of those career development awards, then a NIH Career Development Award for physicians, a KO8, then the RO3, which you can get on top of that to kind of jumpstart your independent research, and then eventually she got an R1. So really beautiful trajectory. She's not only doing basic research as a physician, but also is in charge of some clinical trials, I think in particular in the space of PSC, with some of our local favorites, Pliant, and the other company, I think it's called High Tide or something like that. I don't know where they're from. So a lot of cool stuff going on, bridging basic science all the way to implementation in the clinic. So very excited that you came out here and you share your research with us. Thank you.
I want to start, of course, from thanking Dr. Willenbrink to bring me here and giving me an opportunity to meet with trainees, first of all, and also with esteemed colleagues in the field. So today we'll talk and focus on bile ducts and how we think about bile ducts and how we sort of found and built our own niche. So this is my why, and I'm physician scientist. When I was all my fellowships, including TCT2 at Mayo Clinic in Rochester, Minnesota, I actually worked with one of the kings in cholangiocarcinoma research, Dr. Gregory Gors, and there I focused on patients with PSC. So when I was recruited in the University of Michigan, I was part of liver tumor program right away. I see a lot of patients like this one. It's 47-year-old gentleman with primary sclerosing cholangitis. This patient also has bowel disease, which comes into play with immune-mediated disorders. And he had biliary obstruction and the perihilar mass. What he was diagnosed by us was cholangiocarcinoma. Survival rate is less than 5%. Patients often diagnosed too late to be eligible for transplant. What is dramatic, 2 years ago, he was seen in an outside hospital, and at that time, he had biliary obstruction, bacterial cholangitis, and diagnosed with PSC. So we had 2 years to intervene or diagnose this patient early, but we could not because it's quite difficult disease to diagnose. So focus of my research, one, is how we can prevent this progression from immune-mediated cholangiopathies to cancer. And second, how we in general can improve outcomes in patients with bile duct disorders. So what are cholangiopathies and why we study extrahepatic bile duct, like Helga referred to, and what it is, why extrahepatic bile duct. So I think my animation doesn't work, but I will, just keep going. And how we study it using in vivo models as well as ex vivo models and archived human tissue. I will tell you a story about hedgehog signaling and immune-mediated regulation of bile duct responses to injury. And a little bit, it's the first time I show this very, very fresh project that I want to share with you just to get your feedback. So first things first, let's get to histology and anatomy. Hepatobiliary system, when we think about it, what do you think first? Liver. But it's not just liver, it's also bile ducts. In fact, bile ducts subdivided on intrahepatic bile ducts. They come up to second-degree bile ducts and extrahepatic bile ducts. For large duct and extrahepatic bile ducts, on the right, you see human bile duct histology. We have two compartments of epithelial cells, biliary epithelial cells facing lumen, and underlying it, peribiliary glands, which we think is home for biliary progenitor cells. But we don't know very well what progenitor cells in bile ducts are. And stromal compartment, when someone commented to me that actually, bile ducts is just a tube with few cell compartments, and it's not as beautiful as a liver, not as complex as a gut, for example. So let's see if I will prove them a little bit wrong. So it's a complex tissue. What are cholangiopathies? Classic cholangiopathy is primary sclerosing cholangitis. So this is immune-mediated disorder. It's rare disorder, in the north of Europe, it can be up to 100% per 100,000 population. Males have a higher frequency of this disease. And I want to emphasize, half of the patients diagnosed with this disorder before 40 years old. There are some risk factors identified in a milieu of inflammation, and 80% of patients have also inflammatory bowel disease. These disorders have quite complex pathology, and when you look at this bile duct, it certainly looks very beaten up, and bile duct represented, it's hard to, I don't have, oh, maybe I do actually, yeah. So immune cell infiltration, it does have fibrosis, it's what we call this classic onion skinning, and it also has associated biliary hyperproliferation, which we call ductal reaction in a large bile ducts. What is interesting, we don't have name for it in an extrahepatic bile ducts, but this is most important punchline. There are no effective treatments for these disorders. 60% of transplants in the United States are coming from these patients with primary sclerosing cholangitis because of fibrosis, cirrhosis, some of them also for cancer. And 10% of patients with PSC will develop cancer. Not might, they will develop cancer. And look again, these patients are younger than 40 years old. 5-year survival rates and less than 10%. So what are the gap in knowledge for these disorders? We really did under-recognize difference between intra and extrahepatic bile ducts, which have different histology, they have different embryonic origin, and they have different disease biologies affecting them. We also don't understand extrahepatic bile duct disease pathogenesis coming from first, and we don't have effective models to treat it. Therefore, when I came to University of Michigan from Mayo, I chose to focus not on a cancer, and I was trained as cancer biology, but on a step preceding cancer development and mechanisms of bile duct repair. I focused on biliary proliferation because it's an endpoint that can result in tissue regeneration, but it also can result in cancer when it's uncontrolled. And we developed in my lab, I just spoke with Dr. Maher, we like to tinker. We like developing new tools, and we generated extrahepatic bile duct organoid biobank. We use different mouse models, and we have human tissue biobank. So why is it important to focus on extrahepatic bile duct? Because biology is very different with small bile ducts. This is subsegmental duct primary sclerosing cholangitis. We usually diagnose it only on biopsy. You cannot see it on cholangiogram. And it's very low to none risk of progression for cancer. And some might argue it's just because patients with small duct PSC will transition to large duct PSC. But when you look at large duct PSC, which subdivided further into intrahepatic portion, and it's first degree bile duct, and extrahepatic. So again, back to basic, what is perihilar area? It's from second degree of bile ducts coming right there to insertion of cystic duct, and extrahepatic or distal extrahepatic bile duct, we also call it. It comes from insertion of cystic duct to ample of water. These patients behave very different, and median time to death anywhere from 10 to 21 years after time of diagnosis. Why it's also important? Because cancer is a cause of death in these patients up to 44%. Isn't that terrible? There are risks for other malignancies as well. And look at that. What is the most common site of cancer development in patients with PSC? It's a perihilar bile duct, which we don't study. In fact, majority of epidemiological studies or even TCGA database actually lump together perihilar bile ducts with intrahepatic bile ducts, which it's not, and for cancer. What is also clinically important for me when patient comes to the clinic, a lot of patients, some study here at slide shows 26%, but up to 50% of patients diagnosed in cancer within first 2 years of their presentation, just like my patient I shared with you. When it comes for cancer biology, every different part of biliary tree has different outcomes and has very different flavor. When we look at intrahepatic cholangiocarcinoma, we have FGF fusions, IDH1/2 mutations. They are highly targetable. In fact, they are exclusive for intrahepatic cholangiocarcinoma. You will not see these mutations in extrahepatic bile duct cholangiocarcinoma. But all of the cancer have KRAS mutation and EGFR, and we have work on it, which I don't show today, but also interesting data showing in EGF signaling response in repair. So I hope I convinced you that intrahepatic and extrahepatic bile ducts are not the same and that we should study them. So how can we do it? This is, overall view how we do it in my lab. So we use different mouse models, genetically engineered. We use a lot of Cre drivers, and we target different cells in a bile duct. We also have collection of human samples, and we have prospectively, also generating a biobank of archived, paraffin-embedded tissue organoids. We also use this model to study hyperproliferation that I will introduce to you on the next slide. Usual, histology, primary cultures, we also recognize that organoids, it's epithelial cells, and we use a lot of organoid research in our program. Thus, we also generate primary stromal cells, such as fibroblasts. We also learn to isolate neutrophils from human and mice, and we use biliary explants. So what is model to study extrahepatic bile duct proliferation? This is acute injury model. And on the left, it's a structure of mouse liver. A lot of you know that. On a photograph on the left, this is mouse bile duct. It's less than 2 millimeters, 1 millimeter in diameter. And when we isolate it and ligate it actually as close to sphincter of Oddi as we can, animals developed extrahepatic bile duct dilatation, gallbladder distension, and cholestasis. We were asked many times, can we make it as a chronic model? So how do we do it? As I mentioned, that's very distal ligation of the bile duct and with distended gallbladder. On the right, it's how most of the models were reported. I want to thank Dr. Maher to having discussion with me during one of the liver meetings through SLAM, when I asked, like, what's happening? We just cannot keep this mice alive, and people are reporting, 12 leaks, and that they can keep animals going, and we cannot. What do we do wrong? And she said, we have a lot of, I done it, of course, and we observed gallbladder rupture. And it's what we saw, because we even have the saying, some of the animals, when it's not ruptured, you cannot touch gallbladder because you got it bursted if you're not wearing mask or glasses in your face. So, and we did statistics. So over 72 hours, our animals from 19 dropped to, living 6. So this is ruptured gallbladder, and this is how intact gallbladder looks like, and intact gallbladder after surgery, and this is what we usually see. So we really cannot make this model chronic, but it serves us so well for study what we study. And again, I want to emphasize for trainees, we talked that one of my mentors said, assume nothing. So when we dig through literature, so while this is model we use, we call it distal BDL. This is model that is usually used in the literature. So pay attention to details and see how it's done in other groups. So, and why does it serve us what we want to learn? So when we ligate bile duct distally, this is, so cholangiocytes are very mitotically dormant actually. So don't proliferate fast, and at 12 hours, so green cells through my talk are going to be EDU or proliferation marker cells. And in 24 hours, this is what this animal developed. They developed 70–80% of cells going into mitosis and proliferation, which subsides within 48 hours. So it's very dramatic model. It's actually very specific to epithelial cells and not necessarily stromal cells. We took this model forward and asked question, what are the mechanisms that really help us to, develop this dramatic response right away? Everything should be there because it's just too little time to initiate, many, transcriptional programs, except, acute inflammatory reaction, for example. And I thought about hedgehog signaling. When I was trained on TCT2 grant at Mayo Clinic, I learned about this pathway and its contribution to cholangiocarcinoma. And it's quite complex pathway with 2 receptor system, several ligands which are tissue specific, and a factor which is a transcription factor, GLIS. And when we looked at hedgehog signaling, which is important for cancer, and we thought, well, we know it's some other tissues regulates also proliferation. How does it look in our tissue? So we saw upregulation of Indian hedgehog ligand, and we actually first to show in extrahepatic bile ducts that it's Indian hedgehog and not sonic hedgehogs that everyone studies or talks about. We had no sonic hedgehog in our tissue. And we showed upregulation even of transcriptional targets, PH1 and BLE1. We also learned, because this tissue was new for everyone, where is what? And on the left, in situ hybridization, just showing that actually our ligand mapped to epithelial cells, while our target genes mapped to fibroblasts. Just to orient you to our tissue, so through my talk, so areas that showing glands, it's usually epithelial cells. So epithelial cells, and we often through the talks will map lumen with asterisks. So what we found here is that we actually, cholangiocytes in injury signal to stromal cells. And then what? Does it contribute to proliferation functionally? In my group, we used 2 animal models. One of them was, I don't know what went wrong with PowerPoint. I will just keep going. So on the left, a genetic model with GLI1 knockout, which is a factor of hedgehog signaling. And the right, it's pharmacologic model. And when we use this model, we show that hedgehog signaling not just upregulated, but functionally contributes to proliferation after injury. is a direct effect. And it's very simple. We done this test probably 6 years ago. So, just very micro arrays. So sometimes don't ignore this, simple things. And at the time, it wasn't time of right now, instead of PCR, we just do RNA-seq, but it was a while ago. And CXCL1 popped up. I am not an immunologist. And when I look about, data, I was like, I don't want to, well, I need to learn about the thing because it was coming up in different analysis as well. And we checked for CXCL1 and showed that it secreted, for one, it secreted factor, right one? Okay, so actually better with this one, I think. I thought, but maybe I'm not. Nope, it's nowhere to find. Okay. So thank you so much. So what we show, so green is CXCL1, bile ducts again for, throughout talk orientation, asterisk is always lumen. And it's secreted factor. So here it doesn't show where it comes from. It just shows if it's up, down. So in this particular slide, what we show is that after bile duct ligation, it's enormous increase in CXCL1. And what CXCL1 is, it's a neutrophil chemoattractant. And when we functionally, again, trying to connect hedgehog signaling with CXCL1, we show that when we knock out CLE1 genetically or use inhibitory model, our CXCL1 expression goes down, which is very obvious on the right panels. Well, we already starting filling out, but what the cells are. This analysis was done after our paper was published, but it's all comes together. So when we conducted bulk RNA-seq analysis of sham and bile duct ligated extrahepatic bile ducts, we show significant influx actually or upregulation immune cell markers. And we asked, okay, neutrophils, we were bringing in animal model, and I just wanted to share you to think broader beyond BDL. So this is extrahepatic bile ducts of animals with systemic inflammation. So these animals actually had crushed injury of sciatica. We got these animals because our collaborator next door had this mouse with labeled neutrophils. And we just, it's how it works in my group. You don't need bile ducts or liver. We will get it and we will examine what can we use this model for our tissue. And what we found is that when animals have systemic inflammation, they have enormous influx of neutrophils labeled here in red into bile ducts, which is relevant to human cholangiopathies beyond bile duct ligation. So, and sure enough, we used our inhibitory, tested our inhibitory paradigm with Lys6G antibodies and showed that for one, we can downregulate recruitment of immune cells in the bile ducts marked here. And more importantly, we linked it to the function. So when there are less neutrophils in a tissue, there is also less biliary proliferation. I will probably go faster to this slide. So testing again, and it's like one of my mentor, Gregors, used to say, when you want to test something, you see a fire, test it from different, look at different windows. I don't know if you have to do it in real life, but in science do. So here we interrupt interactions between CXCL1 and its receptor CXCR2, and show when we once again in red it's neutrophils. CXCL1 does regulate recruitment of neutrophils into bile ducts, and it is associated with decreased biliary proliferation. So when we put together this story, we showed that injured cholangiocytes, they do signal directly to fibroblasts, but fibroblasts are not necessarily the ones that signal back to epithelial cells in this particular story. They recruit neutrophils. And also it tells us how important neutrophils are. So we always think about them as something doing damage, but neutrophils are bigger than that. And again, proliferation can be relevant to cancer and as well as regeneration. So anyhow, it's all mouse work, but what about humans, right? So we used our biobank of tissues and showed that patients with without dysplasia and PSC have CXCL1 expression, just like in our mouse model. If you remember this green, protein and a stroma. Same is true with PSC with dysplasia, and moreover, it's the same pattern of expression in extrahepatic cholangiocarcinoma. Another data that we didn't have at the time, but made us think, about bile duct in PSC as a chronic wound in a sense. So this is large bile duct. It's why you see a little bit of liver around. It's perihilar bile ducts. And just to orient everyone, this is artery, vein, and bile ducts. We call it portal triad in a sense. But it's not, it's for large bile ducts. They travel all together. So look at these bile ducts. You can almost feel like it's a hole in a bile duct in PSC, right? So, and it's all actually, so what these cells are, this is spatial transcriptomics on this sample. And for one, it looks beautiful, right, for scientists. And it's also like, it's almost standoff between, damaged bile ducts and the rest of the liver, is it trying to, contain it, or what is going on? But we focused on a couple clusters. One of them, the most prominent cluster, and there is no surprise, cluster 1 in blue, extracellular matrix reorganization, collagen, activation of immune response. But look at this green cluster and these, cells or processes that making hole in our bile ducts in patients with chronic angiopathy. It's all immune innate response, neutrophil migration, neutrophil activation. So which brought us to conclusion that biliary proliferation regulated by, truly very complex, crosstalk between epithelial, stromal, and immune cells. There is also, we discovered this new hedgehog CXCL1, CXCR2 axis in regulation of epithelial cell proliferation. I left it open as epithelial cells because actually it's relevant to other disorders that follow immune-mediated pathogenesis, such as pulmonary fibrosis and, kidney diseases. So it's beyond, biliary epithelium. And we confirm that neutrophils do play actually important role in extrahepatic bile duct responses to injury. And of course, it cannot be one pathway, right? Our body is much smarter than that. So what are other mechanisms? And here I wanted to show you tools that we use in our research as a, also vehicle to tell you a story. So in my group, we actually, when I was recruited, and one of the first things I did, because I was so fascinated, it was this LGR5 papers that come out for generation. It was organoid research was just picking up. And I worked with my mentors group who used organoids actually from primary cells in the gut. And I thought, well, this would be a really important tool to study the tissues that I want to study. And in my lab, we developed organoids from adult mouse extrahepatic bile duct. This is not iPSC models. It's not reprogrammed. It comes from tissue itself, which is already in adult animals. So how we do it, we collect bile ducts from human, mice, we dissociate it, plate and matrigel with presence of growth factors, and they grow just fine. We also collect samples from humans through endoscopic cholangiogram. And as a physician, it's one of the ways that I really appreciate access to. And so generation of biliary organoids from the bile was actually pioneered by Dada Soroka and Jim Boyer's lab, and we expanded on it and started generating organoids also from biliary brushings, also from biopsies, and it's how it all looks like. It's a little brush going into patients' bile ducts when they need to be sampled for cancer, for example. And this is our biopsy tissues, and they also, and we can generate organoids from the samples, and recovery rate is close to 90%. Right now, for generation of cultures, we can freeze them, we can, use them longitudinally. And what is important to know about organoids we generate, it do have polarity, and they express cilia, marked here with alpha antitubulin, and it's these little things, red dots. And for organoids that we generate, lumen is inside, so apical side facing lumen. So this is tool we use for next project, actually, we noticed that in our bulk RNA-seq that a lot of genes that were coming up as a highly upregulated genes, they belong to actually Wnt signaling. In other pathways, that is 26 ligands and multiple co-receptors. And another reason why we thought about Wnt signaling is because for organoids, so organoids, they represent regenerative model, right? And Wnt ligands or activators, they often use for organoids propagation. And we kept asking ourselves, do organoids really need it all? We just use semi-established protocols, used prior knowledge in the field, but that is what was coming from. So, but we confirmed actually that it neutrophil chemotaxis again, and there are a lot of Wnt targets. In fact, it was two of them along ligands that were most attractive to us. One is Wnt4, which is non-canonical, and Wnt7b. And for Wnt7b, when we mapped expression of Wnt7b, it's paralleled completely development of proliferative response in our tissues. So we went after Wnt ligands in our bile duct ligation model and used C59, which is inhibitor of its polymetallization of Wnt ligands, so they can leave the cell. So when we inhibit it, we inhibit expression of ligands from any cells in a mouse. And this is expected increased proliferation after bile duct ligation. But on the right, when we use C59 inhibitor of Wnt ligands, in a sense, we have significant decrease in biliary proliferation. But is it Wnt7b? And it's another tip for trainees, actually. So we hear it a lot that everyone was, warning each other that recombinant Wnt ligands, they actually don't work well because they require very specific post-translational modifications. They should have particular conformation to work in tissues. And we bought this recombinant ligands and they didn't work. So what we did, we went back and we generated our own tools using lentivirus. And right now you're welcome to use this if you want, but we have L cells expressing Wnt7b. And when we used this, not recombinant, but Wnt7b collected from cells from supernatant, we showed that Wnt7b has direct effect on biliary organoid proliferation. Once again, we use C59 just to cover everything, besides 7b, and we showed not in this data that human and mouse organoids, they expressed multiple Wnt ligands actually. And we showed that it does inhibit proliferation. When we use C59, we had no Wnt signaling support in the media. So for one, it tells us that organoids can actually grow without exogenous Wnt stimulation. We could propagate them, and if you're interested, we have it in this paper. And we also use IWR1, which is inhibitor of beta-catenin, which is a mediator of canonical Wnt signaling. It showed that it's likely in a, we didn't look on canonical signaling in this particular work, but we know that canonical certainly contributes to biliary proliferation. But everything is not as simple. So when we think about organoids, you see how proliferating they are, right? So EDU, once again, in green, it's proliferating cells. But what did we discuss a few slides ago? At baseline is, biliary tissue and our cholangiocytes proliferating or dormant. They're metatically dormant. So it truly does not represent this, quiescent cholangiocytes at baseline. And this is very different from gut and from group I work. Gut sloughs off cells every 7 days. So this is normal for gut organoids, but this is not, or epithelial cells, right? But this is not normal for biliary tissue. Thinking about it, we start, and it's certainly, proliferation, just like an injury bile ducts, right? So we start thinking about model, what can we use to model these quiescent cholangiocytes? And we come up with this explant culture. Dr. Becky Wells' group also tried to use them for, answer questions in their research. And they usually used embryonic and postnatal bile ducts. So, and we isolated, once again, bile ducts from mice at baseline and showed that we can keep these organoids quiescent. So green proliferating cells, there is still a little bit more than 1% in a native animal, but it's very, very low proliferation rate. And when we used a Wnt activator called KIR in explant, we showed that Wnt, when Wnt is activated or added exogenously, actually acts specifically on epithelial cells. So epithelial cells are truly a target for Wnt signaling, at least in the early stages. What is interesting, so when you look on the top, this is our explant, so it's all ex vivo, and on the bottom is mouse extrahepatic bile duct after bile duct ligation. So we saw that we got what we wanted to get. So summary 2, it seems that developmental pathways, and I don't want to leave it just to hedgehog or Wnt. I think we need to leave it broad. Developmental pathways, they certainly regulate, extrahepatic bile duct regeneration, and I will limit it here for, obstructive injury because it's what we study. So it's what reviewers ask us all the time, and it's what we ask ourselves all the time. How can we study chronic cholangiopathies of, extrahepatic bile ducts? And we, of course, went to the models that are established, published. One of them is DDC diet, as well as MDR2 knockout animals. And when we examined, and first we look, of course, for proliferation, because again, as a physician scientist, I was stubbornly, focused on proliferation. But we couldn't find many green cells, and tissue didn't look so different. And DDC diet extrahepatic bile ducts, so MDRs, and it's, we took it to next step to follow up on it. What is interesting is that bile duct ligation just gave us our perfect biliary proliferation model, but not established models of chronic cholestasis. And next, we did what we usually do in my lab. We start looking at how, particular intervention or model looks in our tissue of interest. So assume nothing, check your tissue. So how does DDC diet work? It interferes with heme synthesis and blocks this enzyme called SPEDGE. So there is accumulation of protoporphyrin. And in the liver, what happens, and I will show you a slide, a little bit further, that there is accumulation of this protoporphyrin or porphyrins in a small bile ducts. This model associated with liver inflammation, ductal reaction, fibrosis. So it's really models component of the liver disease. It's not necessarily models, human biology, but it models aspects of disease. But what do we have in extrahepatic bile ducts? So this is a new project for us, so bear with me, I'm thinking through. And we done it for 4 weeks. It's actually, animals are quite sick on this diet. So 4 weeks, we probably in the future will go to 3. But in the liver, and here we use liver as a control. So I'm not showing you, extrahepatic bile duct first, but liver is a control because it's well-established model, is published, so many groups using it. So we have plaques as expected. We do have fibrosis and ductal reaction in all these portal triad areas, but bile ducts or gallbladders, they don't look that much different actually. If you, pay attention to details, you might see that there is a little bit, thickening of the wall, but not much dramatic changes. We also checked where is this protoporphyrin is, and we used its properties that actually autofluoresce. And we checked it. So in the liver, we know we have this plaque. So purple is porphyrin plaque, so porphyrin, product. And this is gallbladder, and there is some, porphyrin presence there. It's getting stuck in a cystic duct, actually, and not much of porphyrin in extrahepatic bile duct. So once again, we did what we do. We characterized our tissue and then bulk RNA-seq on different regions of the biliary tree in animals treated with chow diet or DDC diet. When we did principal component analysis, our samples grouped mainly by tissue of interest. There were genes certainly differentially expressed in bile duct and the gallbladder, not at the degree as the liver. But what are these genes are? So there is certainly increase in epithelial cell markers in the liver, bile duct, and biliary tree. It's not as consistent in the gallbladder, and it's mainly in a setting of hyperplasia, because when we done EDU and we do very short term, so it might not be a good way to check it for this model. Still on DDC diet, even short term EDU administration for 2 hours had a lot of positive cells, but that was not true for extrahepatic bile duct or gallbladder. What about inflammation? So nice, blue and red in the liver, but not so true in the bile duct and in the gallbladder, right? Although, common, pretty much important contributors, and with asterisks, we have statistically significantly differentially expressed genes are TNF alpha, for example, MIF, IL-18s, they were differentially expressed. And there is also difference between extrahepatic bile duct and gallbladder. But as you look back, cystic duct, which is exit from gallbladder, is, plugged by the porphyrins. So does it play a role? Perhaps. So next, we, of course, check on the presence of immune cells based on our interest, and, it's pan-immune cells marker. That and the liver here is on the right. So we do have an increase of expression of some inflammatory markers, neutrophil increase in liver, but in our tissue of interest, it's not just doesn't change, it's went down. So which is probably just because it's a whole tissue, but point is that there is no increase. And as expected, monocyte macrophage markers are upregulated in the liver, but not in the bile ducts or in a gallbladder. So this is next. We always look in fibrosis because this DDC model, it's a classic model of, cirrhosis and liver fibrosis. Once again, it's serious red, marking collagens. And here are the plaques and DDC duct, no plaques. And so at baseline, we need to remember there is a lot of collagen in extrahepatic bile duct or gallbladder baseline, which actually makes it difficult to study for fibrosis endpoints, because how do you look for it, right? In the liver, we'll do serious red, trichrome, and Mason trichome, but what do you do in the bile ducts? So, and here we show that there is some thickening of the, stroma once again, but there is more like rarefication, and it's not necessarily, collagen proteins, it's non-collagen proteins. And this is gallbladder. So we didn't find in the bile ducts that many changes, and not as dramatic changes as in the liver. And we ask ourselves, how does bile duct can potentially protect itself from injury? And we did, pretty much, we pulled top differentially expressed genes, and a lot of them, I think, are very interesting. We do have mucosal barrier genes. CHIL3, glutathione-related genes, MARCO, HMOX, it's a heme synthesis, which is pertinent because it's DDC diet, it's what it does, right? And also MAC1, very interesting protein, and there are right now, this protein this is gene, but protein encoded by this gene was shown to contribute to cancer development, actually, and contribute to cancer progression. There is some vaccine developed targeting it. So it seems that a lot of things, point towards, and a GSA analysis showed us that a lot of genes are coming from NRF2. It's nuclear factor erythroid 2 related factor 2. It's pretty much defense mechanism against oxidative stress. On one hand, we're glad that it was such a strong signature, right? So like majority of us, our, differentially expressed pathways or genes that belong to this family. But, in the liver, it was already shown that besides, increased inflammation, and so NRF2 is also upregulated and might protect from injury as well. This is NRF target genes, and we wanted to see if it's direct effect on epithelial cells, and we designed this experiment when we used organoid models and treated organoids with protoporphyrin. What we found was quite interesting. It's not what we expected. We looked for genes, but I think it's biologically, it's interesting observation. So on the top, it's vehicle-treated organoid. This is our organoids, ecadherin. It labels epithelial cells. And PPIX, we used once again, its autofluorescent property. And we showed that enigmatically, actually, so basolateral membrane on our organoids is outside, right? So epithelial cells, they take it up from basolateral membrane side and transport it into organoid lumen. So they're not just, sort of passive with that. They probably want to extrude it, get rid of it, So that was quite interesting. And this work was done with extrahepatic organoids. And because we always focused on extrahepatic and large perihilar bile ducts, this is something new and cool for us. This data, we can have all. So we start playing with intrahepatic biliary tree and generate organoids from intrahepatic biliary tree. So, and it's a very fresh culture. And for this is, map where we got it from. And this is small intrahepatic bile duct, large, and our usual extrahepatic bile duct and gallbladder organoids. Summary 3, extrahepatic bile ducts and gallbladder are relatively protected actually from DDC diet-induced injury, and how they do it, probably developing this massive response with NRF2 antioxidative response, and also upregulation of genes related to mucosal barrier. And, this observation that, bile duct, it's not bystander in responses to DDC diet injury. It might, help to, sort of get rid of this, toxins that causes phenotype. And of course, distal bile duct ligation, which is short-term, cannot be last long, is still best way to study proliferation. Getting back to bedside, this is my another patient that I saw in the clinic 2 weeks ago, actually. 63-year-old gentleman with bacterial cholangitis. We diagnosed him with PSC. And just to show you how tricky this disease is. So this is human cholangiogram. We use MRI sequences to build biliary tree based on some physical properties. And this is gallbladder of the human. This is common bile duct, and this is interhepatic biliary tree. So when you look for it, it looks all right. But when you look from another side, there is a disconnect between bile ducts. So we are very much concerned that our patient is about, or, again, remembering that first 50%, the 50% patient will be diagnosed with cancer in the first 1-2 years, we are really concerned about cancer. How do I take my observations from the bench to bedside? So I actually, we always follow patients with PSC extremely closely. They have all our phone numbers. Since my times at Mayo, I had this QI project as a fellow when we call it antibiotic in the pocket. So I want my patient to be started on antibiotics right away. And my research supported this approach because we want to control infection as early as possible. We don't want these neutrophils to come in and, stimulate this hyperproliferation. But also, can we take this research and think how we can harness, contribution of this developmental pathway to regeneration? Can we use some components of it and knowledge, and also tools, I shall say, like an explant to study, how bile ducts can be preserved for liver transplantation? Because bile duct actually ischemia is one of the Achilles heels of liver transplantation. And we also should consider contribution of this developmental pathway to cancer, because once again, when regeneration goes uncontrolled and unstopped and propagates endlessly in these patients with chronic inflammation, it can turn into cancer. In a summary, regulation of extrahepatic bile duct Responses to injury involves complex crosstalks. When we use models, we need to ask, first of all, if these models, they work in our tissue and what is phenotype in our tissue. And we need to understand, because models are not perfect, but they're good to model particular aspects of the disease. Like bile duct ligation is good for proliferation, but DDC, MDR are not, but they might be good to study mucosal barriers. So keeping that in mind. And of course, I believe that if we understand biology of extrahepatic bile duct responses to injury better, we can help our patients. I want to thank my team, and it's my sister lab also here, and our funding. And Holger saw the slide. My daughter put it together for my June conference, but this is bonsai tree in Michigan, University of Michigan bonsai garden. And she took it for me because it reminded her liver. And she knows that I'm obsessed with liver. And what she did for me, she put that. And I think through collaborations and with patients, scientists, and clinicians, we really can build this, very strong trunk and very strong biliary tree, so we keep liver healthier. Thank you.